In this topic, we start our study of Deuterostomes, which includes the Echinoderms and the Chordates, which is a group we belong to. And so we're going to start with section 1, which covers the phylum Echinodermata, or Echinoderms. And Echinos means spiny, and derm is skin, so literally translated as spiny-skinned animals. They're all exclusively marine. And there are deuterostomes, so they have deuterostome development, radial cleavage, for example, indeterminate cleavage. Their second opening becomes the mouth. It's in the name, stoma. They have an endoskeleton, and so that means the skeleton is underneath their epidermis, or deeper than that. And they have pentaradial symmetry. So there's a radial symmetry here. It's kind of odd because we're supposed to have bilateral symmetry, because we're in the bilateria. So the deuterostomes are out here on this cladogram here. And here are the chordates, but we're going to cover the echinoderms. And while they have radial symmetry, it's a pentaradial. You can see this the blue starfish up here in the top diagram. 1, 2, 3, 4, 5. And they have a mouth underneath, and the other side is aboral. So when you're radially symmetrical, there's no anterior, posterior. So how could they be with the bilateria? Well, they evolved to go through metamorphosis to have this radial symmetry, but their larval stage is still bilaterally symmetrical. So their development gives clues that they do belong to the deuterostomes and the broader bilateria. This group includes the sea stars, brittle stars, sea urchins, which you may have heard of, starfish before. That's sea stars, sand dollars, and sea cucumbers, which are kind of odd. So the learning outcomes for this one is to list the specific characteristics for echinoderms, then explain what is meant by pentaradial, which kind of covered that right now, and then describe 5 extant classes of echinoderms. There's many groups that have gone extinct already. So the origins of echinoderms appear nearly 600 million years ago during the Cambrian explosion. So all the way back then. Now the origin is unclear, but they did evolve from bilaterally symmetrical ancestors. And the evidence is seen in their larvae. Here is a starfish larva here, image from your textbook. From a sea star by the name of Asterias rubens, and it is bilaterally symmetrical. There's a mouth here, and you can see the gut here, but you can only cut it along one plane and get a mirror image. If I cut it this way, you don't get a mirror image for it. So the symmetry of this group is radial and their endoskeleton. So when the adult develops from the bilateral larva, is going to be pentaradial, and so the radial symmetry, as I mentioned, there's no such thing as a left or a right side, a dorsal or ventral. Instead, you're going to have the oral, and then the opposite side is the aboral side for it. So now all of their organ systems that are within there are going to branch out in this radial sort of symmetry. Even their nervous system is going to consist of a ring, and then branches of a nerve going out through to each of the arms of a starfish, for example. So the interesting thing is there's no centralized nervous system like we're used to when we talk about a brain. But these animals are still capable of very complex behaviors. Pretty interesting. When it comes to the endoskeleton, the epidermis covers it. This is not like the exoskeleton of an arthropod, which we just covered. That skeleton is produced by tissues underneath and is totally on the outside, and it's not made of any living kind of cells overall. So that's what's meant. And the skeleton is actually made of pieces which may or may not be fused together. In a starfish, these ossicles, which are made of calcium carbonate. And so that's something we've seen before. This compound here fit together they may be fused. If you watch the Shape of Life video, you'll see how the ossicles and the muscles underneath allow the starfish to move in quite complex ways. But here's underneath the epidermis on the outside. This is just a section through one of the arms of a starfish. You can see those ossicles right there, right underneath the skin. So again, they may be tightly or loosely joined, depends overall. And their skin have this mutable collagenous tissue, which can change from rough to rubbery to weakened fluid overall. And their skin does have these spiny structures protruding from them, made of that calcium carbonate, giving them their name, the spiny skin animals. So they have a water vascular system, and so they have a system that, again, follows that pentaradial structure. So just like the nerve ring they have a circular canal. And from that circular canal, underneath these green structures here in the starfish that we see right here, they have the stomachs there, and underneath there's a yellow ring underneath. And then this radiates outward, the canals, and those canals that radiate outward are called the radial canals. And what happens here is the water enters from a sieve plate. It's also called a madreporite, which is this term right here, madreporite, or a sieve, which is sort of a filter. So there's little tiny pores in there, so the water's sort of filtered, and then it goes down through a structure called a stone canal, and then from there throughout the rest of the water vascular system. So again, it's radially organized, and those canals extend from the ring outward, from the ring canal. And the stone canal I mentioned, that comes from the madreporite, or sieve plate, down to the ring canal. Now what happens here is that water system will actually fill up these tube feet here, and the tube feet actually stick out on the bottom side. You can see some of the tube feet there underneath in the drawing here, and then here's an actual real picture of an arm of a starfish, and there's the tube feet there with little suckers there. So basically they can pump water into those feet, and that causes those feet to stretch out, and then muscle can swing those feet back and forth in a coordinated way. The sea star can move in any direction it wants to, and the structure that pushes that water down, as that water goes to the radial canal, it feeds each of these little bulb-like structures that are there, and the bulb-like structure is the top of the tube foot, and that bulb structure is called an ampulla, or ampullae would be plural. And that pushes the water down in there. And so the tube feet are used for movement, locomotion. They can also be used for gas exchange. Animals need oxygen, right? And for feeding. Starfish is very good at hunting bivalves. Remember the mollusks, the bivalves with the two clamps. So what they can do is they can find like a mussel, which is a kind of like clam, and then go and wrap their arms around it and then pull. There's a real good video of this in the Shape of Life video that I have as a supplemental link. And they pull and pull and pull until the muscles of the bivalve, the adductor muscles, get tired and fatigued. And then they're able to get their stomach in there. They have two stomachs. They have a cardiac stomach and a pyloric stomach. And they actually will extend one of the stomachs into the clam itself, and then just begin digesting the clam while it's still alive. It's pretty gross, or cool if you think about it. Many of these echinoderms do have some ability for regeneration. So if they lose an arm, they can grow back that arm. And some can actually asexually reproduce by breaking off a piece, and then that piece becomes a whole other individual. During sexual reproduction, they are gonachoric, which means there's a separate male and female. Gametes are going to be released in the water, and then when the sperm fertilizes the egg, the zygote develops to embryo, and then eventually a larva, which is free swimming. And each class is going to have a characteristic larval type, but the larval type, remember, is bilateral. Has bilateral symmetry. Okay. And so if we look at the fossil record suggests that there were more groups of echinoderms, but only 5 are extant or living today. And we're going to do just a brief discussion of each one of those. And so there were 20 classes, and again, 5 are alive today. There's that class Asteroidea. And you're going to have to know these terms for the second half of the lab when it comes to the lab exam. Those are the sea stars and daisies. And there's a picture right here of some sea stars. And then the Holothuroidea, those are the sea cucumbers. These are a real odd group. You'll learn more about them through the lab. It's kind of like a sea star, but instead of the mouth facing down, faces forward, and they go around vacuuming up the bottom of the ocean. And from that substrate, they can absorb organic material as their nutritional source. And then the Echinoidea, which the class translates to pretty spiny, and that's the sea urchins and sand dollars. And there's a sea urchin there. You can see how it looks real spiny. And then the Crinoidea, the class Crinoidea, those are sea lilies and feather stars. And there's one right there, a crinoid. And then the Ophiuroidea, this one are interesting. They're called brittle stars because their arms can break off quite easily. But they're interesting, and the name actually translates to snake. Ophio means snake, is Greek origin. And they don't have 2 feet for locomotion. Instead, they wiggle around their arms, and the arms wiggle around looking like a snake-like movement. So looking at the class Asteroidea, which are the sea stars and sea daisies, the same picture from earlier. There's at least 1,500 known or identified species, and they are predators. Like I said, they like to go after certain kinds of shellfish like clams and marine gastropods. They can be found in deep oceans and intertidal zones. An intertidal zone would be one where between high tide and low tide, the moon goes around the earth and it pulls the oceans with it, and so the water levels rise and go down twice every day. And in places where there's rocky shores, pools of water get built up, and you can find sea stars within there looking for food. For these guys, this group, their arms taper. They start off wide at the base, and then they taper out and get narrower towards the end. And that's so when you look at a brittle star, that's not the case. They don't do that. And they all merge onto the central disc area there. That's the region of the body there. The body's going to be flattened, and it's going to be covered with a colored or pigmented epidermis. And then the Ophiuroidea, they're commonly called the brittle stars. This is the largest class. C means around, so circa 2,000 species or so. Their arms have a more or less equal diameter all the way around, and their arms can easily autotomize, and that means break off easily. It's almost as if they control it breaking off, kind of like a lizard when they lose their tail, and that's why they're called brittle stars. The 2 feet don't have any ampulla. Remember, the ampulla is what pushes the water down to cause the 2 feet to extend downwards. So their 2 feet are actually used for feeding and not moving. And so the way they move about is how I mentioned. They wiggle their arms around so that the arms look more snake-like, and they can even get up into the water column itself and swim with the movement of those arms. This group, the sea star has a mouth, has an anus on the other side. This group has no anus, so through evolution they lost that, and so they have to eliminate any waste through their mouth. Sounds pretty gross. And then here's the Echinoidea. These are sea urchins and sand dollars, and these, after I watched the Shape of Life videos, one of those supplemental videos, I really understood how this looks actually a lot more like a sea star than I first thought. And this is their ossicles, the ossicles underneath those large spines there. The spines have been removed, the skin is no longer there, and neither are the internal organs, but they have 2 feet as well. They have these larger spines that they can actually move with muscle and so on. But picture a sea star with its arms, and then the arms just fold upwards, and then the ossicles under there become fused, and so you can almost see those arms right here. Like there's one arm right there, there's another one there. And so that's pretty interesting. And then there's the sand dollars that belong to this group. And so picture a sea urchin like the ones we see here, and their skeleton there, and then you just compressed it downward, and what you get is you get a sand dollar. And this is a living sand dollar right here. It's got a pretty colored epidermis there, and their mouth is underneath. So this section is on coverage of the phylum Chordata, or the chordates, and this is the group we belong to. So we look at the cladogram here. We just finished talking about the echinoderms, and we're now looking at the chordates. And these are the two Deuterostome groups to show that Deuterostome development. All those other animals that were bilateral were the protostomes, and that was the prior chapter. So what are our learning outcomes here is to list what are the defining characteristics that all chordates share, because there's going to be some that look nothing like us. But there are some things that they have that ties them to us as far as sharing that common ancestry. And then define the evolutionary relationships of chordates to other taxa. So how are chordates related to echinoderms and related to the other animal groups? So for chordates, they're going to have an endoskeleton, just like the sea stars and the other echinoderms. But it's really a lot more internal than the echinoderms. The echinoderms are more like an exoskeleton with skin on top in a functional sense. It is internal, though. So the endoskeleton of a chordate is truly internal, and that's the point being made here. While the endoskeleton, which means within, rather than the exo of an arthropod, exoskeleton, the endoskeleton is actually more like an arthropod because those ossicles are actually on the outside and the organs are on the inside there. So this group Chordata includes things we are familiar with, like fish, amphibians, reptiles, birds, and mammals. But all of these are actually a subgroup of chordates. These are all vertebrates. It turns out there's other chordates that are not in this list, but we'll see them here in a little bit. So what are the features that define birds, fish, and some real weird things we're going to be seeing right now is that at least at some point in their development, whether it's early in their larval stage or all the way or in their adult form, they're going to have these characteristics at some point in their life history. They're going to have a dorsal hollow nerve cord. All of the nerve cords we've been looking at so far, like for the earthworm and insects, they were along the belly or ventral side. For chordates, the nerve cord is on the dorsal side. So you can see here the brain is developing and eyes, which also develop from those structures here. And you can see in this embryo, there's what's going to become the nerve cord, and it's on the dorsal side over here. So this is the dorsal side, this is belly side, this is ventral over here. And so they're going to have a notochord, and a notochord is going to be a flexible but a sturdy, somewhat semi-rigid structure there, and that's here in red, that notochord there. In vertebrates, it gets replaced by the backbone, by the vertebral column of vertebrae that we get. And then we all have pharyngeal slits, and we can see them here in this embryo, chordate embryo. All chordates have this, and in fish, these become the gill arches. In mammals, some of those parts become part of the inner ear, some of the bones of the inner ear, and so, but they all have that same origin. And this gives us a clue of our aquatic ancestry. All animals with backbones, their ancestors started in the water as fish. Okay? And then we got a post-anal tail. And that means there's a tail that comes right after the anus. So we follow this rudimentary digestive system. That's where the anus is right there. That's where waste would come out. And here's the tail. Even humans have a tail. We have a little tailbone. It's a very reduced tail, but we still have that tail. So some other chordate characteristics. Again, at some point in their life history, these animals will have that in there. So what are some other things that distinguish chordates? Well, the chordates are going to have an arrangement of muscles in segments. And these segmented muscles are called somites. And remember, we want to be careful not to say, well, that segmentation was gotten from a common ancestor. Segmentation has arisen in different groups of animals independent of each other. It means we don't get the segmentation from another, but the segmentation has this common theme that one of the things it allows for more flexibility of the body and better and more complex movements, and the opportunity to specialize certain regions there. So we have the chordates are going to have an internal skeleton that muscles can work on as well, like our bones in our body. So here's an image of a mouse embryo. Doesn't look much different than a bird embryo or a dog embryo or a human embryo very early on. But you can see the somites there that are forming. These are the segments of muscle that are starting to form along the trunk of the body there in this 11-day-old embryo. This is the same text from the prior one. I don't know why I left that slide in there. I should have removed it. This one just has that picture. And then again, that internal skeleton that they have here, this is an animal called a lancelet, which means a small blade. A lance is like a blade or like a sword here, and they bury themselves in the water. They're not very big. They're a few centimeters in length, and they filter feed. They take water in and capture food particles in these gills, and then the water comes out the atrial here. But you can see the dorsal nerve cord that's in yellow, and then that purple line is the nodal cord, which is rigid, not extremely rigid, but flexible enough, and muscles act on that, and that allows them to wiggle and swim from one spot to another. They like to bury themselves in the substrate here. But even these have structure that muscles can pull on. For vertebrates, we would have our bones that occurs. This segment is going to cover chordates that do not have vertebrae. So these are the non-vertebrate chordates. And so the phylum Chordata can be divided into 3 subphyla. The non-vertebrates are these first 2 here. The subphylum Urochordata, these are called tunicates or sea squirts, and there's some other bizarre animals that are in there. And then the subphylum Cephalochordata, which are the lancelets that I mentioned in the prior section. There's a Shape of Life video on the chordates, and they talk about some of these really bizarre Urochordates. And then there's the group we belong to. This is the 3rd group. It's called the subphylum Vertebrata. These are the vertebrates, but the formal name, taxon, is Vertebrata. So this section is going to focus on the first 2 there. This is an image of a sea squirt, and sometimes they're called tunicates. The salps are the bizarre ones. There's approximately 2,000 species. They're marine. Their larvae are tadpole-like. Their adult form looks nothing like what we would consider or that we can closely relate to. But the key is looking at their larval stage is really looks a little more familiar to us. The thing about these guys, they are filter feeders. They're sessile. When they become an adult, they settle. The larva settles and then goes through metamorphosis to form these. And they have an in-current and ex-current siphon, so water is taken in through one siphon. And then they have gill structure that common theme. The gills will pick up food particles, and those will get the particles will get moved to their mouth so they can ingest those particles. So these are filter feeders. There's no major body cavity, no segmentation there. And many produce a tunic, which is why they get the name tunicates. Tunic is a covering. And this covering is made of cellulose, which is interesting because the only other place we've seen cellulose has been in the cell walls of plants and in xylem of wood and so on. So here we have an animal that's capable of producing this kind of polysaccharide here. And so here's a picture of one, I guess that goes by the common name sea peach, and then there's the scientific name. Halocynthia aurantium is probably the way it's pronounced. But you can see the 2 siphons there, and then the body here itself look nothing like this. This is what it looks like internally. Water would be taken in here through here, and then the back of the throat of some of these animals would be called the pharynx here. This endostyle is sort of like a basket-like structure where the food is captured. So this is like their gills. So the water comes through here, and then that water passes out the excurrent siphon. Down at the bottom of the endostyle, there's the mouth, and then from the mouth we get into the visceral mass down here. And within there you have your stomach for digestion and the intestine, and then eventually we leave out here and waste products is released here where water's going to be able to sweep it out. The gonads are also released through there as well. Not the gonads, but the gametes produced by the gonads here. So there's a heart in there and so on. But look at their larva. The larva looks more like a tadpole. And then when you look at it, there's your dorsal nerve cord. Here's your notochord, which in vertebrates becomes the backbone. They have those gill slits up here, from the pharyngeal pouches. And then they'll have a postanal tail out here. I'm not sure where the anus is in the diagram, but it probably exits out somewhere around here for their food waste. And then there's the subphylum Cephalochordata. Cephalo means head. I'm not sure the origin of the name, the etymology of it overall. I would have to look up the history of that name. But they're commonly called the lancelets, which means small lances. The notochord stays with them throughout their entire life. They don't build a backbone. They don't look like sea squirts. And they're filter feeders as well. They have no real distinguishing features. However, you can see the segments within the body. And then those white structures that are in there are actually the gonads for these individuals. And they like to bury themselves, bury most of their body in a substrate there and then just filter feed. And then they can jump out of their location and then go on, move to a different location in there. So they do filter feed. They feed on plankton and then they're close. These are probably the closest relatives to the vertebrates if we were to look at that group. So they're called lancelets, and genus is Brachiostoma. So now a brief intro into the third subphylum of the Chordata, and this is the subphylum Vertebrata. These are the vertebrates, or the animals that have backbones. Your learning outcomes for this one are to distinguish between vertebrates and the other chordates. What is it makes this group unique? And then explain how cartilage and bone contribute to increased size in vertebrates. So with the subphylum Vertebrata, the vertebrates, these are chordates with a spinal column of backbones, and they're distinguished from non-vertebrates by that vertebral column. And this vertebral column encloses and protects the dorsal nerve cord. So think about your spinal cord, which runs along your back there. This is a cross-section or transverse section through an embryo. You can see the ectoderm there, and the ectoderm actually starts to fold inward during some of this development and then pinches off and forms the neural tube. And this, remember, the notochord runs the length of the body here. That's a cross-section through the notochord. So in vertebrates, a backbone begins to form or develop in that area where the notochord was. And the vertebrae there's many vertebrae, so you have many vertebrae running down your back. So this is just one of the vertebrae at that area where the section that we're looking inside the body from. And so you can see we start to form here the bones of one of the vertebrae there, and then these would be the ribs beginning to form there. And then eventually you get your neural arch around there, and you can see that a hollow nerve cord that's in there, which is going to become your spinal cord, is protected within a hole that goes through that backbone. And so eventually the notochord disappears on you and is replaced completely by the backbones, the bones of the back. And so we also have a very distinct, well-defined head region with well-developed brain as well. So other things that vertebrates have, a neural crest is a structure of the development. So this neural crest is it's a unique group of embryonic cells. So this is early in development of a vertebrate, and this forms many vertebrate structures in there. And then you have internal organs, very complex, well-formed organs: liver, kidney, glands of the endocrine system, heart, a closed circulatory system, and an endoskeleton is made of both cartilage and bone, and that allows for very powerful muscles and to support a great deal of weight on there. So in this picture, this alligator here, you can see inside internally, you can see the backbone, the vertebrae there, and then they cut away into the backbone so you can see the spinal cord, and you can see the heart and the liver and the digestive system in there. Here's the kidneys for water balance. So this little statement right here is talking about one of those objectives. What's the big deal with this internal skeleton made of cartilage and bone? Good place to put very powerful muscles, and the bones themselves, skeleton can support and protect those vital organs in there. So the history of vertebrates, when we look at them, what was their origin? So we have to go back to the oceans to find some of the first vertebrate fossils. About 545 million years ago, you had these early vertebrates that had a mouth on one end and a fin on the other. They didn't have paired fins like we see in today's modern fish. These early vertebrates didn't even have a jaw, so they had a mouth, but they didn't have like a jawbone. And then jawfish evolved, and then they started becoming more dominant because they're able to use the habitat in ways that the jawless fish could not. Eventually, common ancestor to today's fish and amphibians moved on to land, much like the way plants moved on to land. And so the amphibians invaded land, and then the amphibians, one group of amphibians there was a great diversity of amphibians, but gave rise to reptiles about 300 million years ago. And then within 50 million years, the reptiles replaced and dominated over the amphibians. In fact, some amphibians were starting to move on to become very terrestrial in their lifestyle. Some reptiles gave rise to birds. In fact, birds, the evidence now strongly suggests that birds and dinosaurs and crocodiles share this kinship or close ancestry. And so the mammals began way back with those reptiles at the same time, but the mammals were small and not very much of an influence on the landscape because the reptiles were there. But then at the end of the Cretaceous period, there was a mass extinction. All the dinosaurs disappeared, not all at once, but by 65 million years, you don't see any dinosaurs in any rocks that are younger than that. And that corresponds to a massive meteorite impact asteroid to hit the planet. Actually, here on this hemisphere, they're right near Central America and the Yucatan Peninsula. They're right off into the water. So what we're going to see here when we start to study this history of vertebrates and the diversity we see in that, the history basically is going to be like that of plants. You start out in the water, and then you invade to land, and a series of adaptations that allowed that. Completely moving on to onto land for plants was the evolution of the seed and pollen. Here, the amniotic egg, which prevents the embryo from drying out, was the ultimate clincher for completely moving on to land. And then some move back into the water like the whales. So here's classification and the phylogeny that we see of the vertebrates, starting off with jawless fish out here, which are still alive today, and then ending with, on the other end, mammals and birds and reptiles on the other end. And we're still going to use a traditional classification scheme. We're still going to talk about birds as a separate group, even though birds share a common ancestor with crocodile and dinosaurs. They're actually basically reptiles with feathers that can fly. But we're still going to refer to them as a class called aves. And in that sense, that makes some of these groups paraphyletic, which goes back to how we try to name groups. If you don't include all members that share an ancestor, then your group is paraphyletic. So if we keep the birds out and we just talk about reptiles as separate, then reptiles is a paraphyletic group because it doesn't include a group that should belong with them. The same thing goes with fish here. We're going to see that fish, like these lungfish right here, which are still alive today, are actually more closely related to land animals than they are to some other fish. So it's going to be a pretty interesting exploration that we're going to do with the rest of this chapter. So in this section, we are going to explore our first vertebrate group, our first classes of vertebrates called fishes. And there are several groups of fish. So there's several, so we have the subphylum Vertebrata, and then there are several classes. And throughout this journey that we're going to look at, we're going to be going forward in time, looking at the evolution of one group that still has members alive today, and then we go back again and see that other groups were around and evolving at the same time. And so things can get a little confusing. So one thing that I did, playing the role as a student, was I made a timeline. And it included the timeline that we had with the geological time periods up on the top there. And I just used a regular paper, and each line I think made it represent probably 50 million years. No, I think it was 20 million years. From going from present all the way back to the start of the Cambrian, where the Cambrian explosion was. And every time something significant was mentioned, I added it to there. First, I put the different periods up here, and then I started putting key fossils that were found, and then the range of time when certain groups of organisms started to appear in the fossil record. And then I also put if a whole group went extinct, the line cuts off, and so on. And when I did this, I have this visual reference now that I can go back and say, oh, okay, that's where we're at. And now you can visually see the overlap of these events, because we'll go back to one period, then move forward in time, and then go back again. And so another group was evolving at that time. So I found that this kind of helped me go through the chapter a little better when I tried doing it that way instead of just looking at the book and the notes. So your learning objectives, just a suggestion. Your learning objectives here are to describe major groups of fish, and it's fishes. When you're talking about more than one species, it is grammatically correct to say fishes. And then discuss the significance of the evolutionary innovations in fishes. And so you can see some different fish here. Here's a tuna. This is a sea dragon or a seahorse, and then here's an eel here. That's just some of the diversity of fish that we see out there. It is the most diverse group of vertebrates. Over half of all vertebrates are fishes, and they provide an evolutionary base for the invasion onto land for amphibians. Amphibians, that's the next group to evolve later on. And so looking at the early fishes, we're going to look at these ancestors. They were evolving back during the Ordovician period, which was from 485 to 438 million years ago. And right there, I was able to draw that on my timeline. I put a bar there from that approximate range. And these were jawless fish. These were the ones that just had a mouth and a tail fin, and that's it. They didn't have the paired fins like you see in this tuna here. The tuna has these pectoral fins, and they have a pair of anal fins, and very much like our pelvic fins. Sorry, they have these pairs of fins, and these fins correspond to our limbs. Later on, we'll see how interesting that works out there. And then jawed fish appeared. And they appeared during the Silurian. And that's 438 to 408 million years ago. So right here you can see Ordovician, then we move into the Silurian period there. So what are some characteristics from fish? Fish vary in their appearance. I just looked at the three fish we looked at a while ago in the image. They vary in their habitats. Some are marine, some are freshwater, some live in deep water, some live in the open water, some live near the shore, and so on. But most of them are going to have these characteristics. And there's some exceptions here. The hackfish and the lamprey, those are the jawless fish. They have a rudimentary structure that resembles a vertebral column. So they either have very rudimentary structures that are not quite notochord, not quite backbone, or they have some backbone and then missing some parts of it here. But these are the jawless fishes. So all the other fish are going to have a pretty solid backbone. They're going to have jaws and paired appendages. Again, exception are the hagfish and the lampreys. The hagfish, by the way, are scavengers of the ocean. When something dies, they like to eat the rotting material there. But for other fish, they're going to have jaws, and they're going to have pairs of fins, the pectoral fins and the pelvic fins. Earlier I said anal fins. There are anal fins, but the pelvic fins are paired just like your legs are paired. And when it comes to, there's a group of fish that you're going to be really fascinated with this. When you look at them, they're still alive today. They're called the lopefin fish. And they have this fleshy nubs that stick out of their body, and they have the same bones that you have in your arms, the same arrangement of bones. They're just descent with modification. Those bones get longer and serve other function, like flying in bats and so on. Fish are going to have internal gills, not external gills like we've seen or we may see in other kinds of animals. They're going to have a single loop blood circulation. Other vertebrates we're going to have two loops, two circuits. Fish have just one circuit. Blood goes from heart to the gills to get oxygen, to the body to deliver the oxygen, back to the heart, and then repeat just in one loop overall. And this is rather interesting, is that the evolutionary history traces back the deficiency we have for some amino acids. We have amino acids that make up our proteins that you need to get in your diet. If you don't, you're going to get sick because you won't be able to make proteins because you'll be missing, I think it's 7 essential amino acids. But fish have started it all, and they lost enzymes to produce the aromatic amino acids. So this is aromatic enzymes is a subclass of those 20 amino acids we use. And so all the vertebrates that are alive today inherited that deficiency from what fish have today. And we have even more amino acids that we need to get in our diet. I think it's 7. They're essential in your diet. You get them all by eating beans and rice, by the way. So here's a table of some fish. Some are extant and living, and some are not with us anymore. And just like any other table, this is much better once you've gone through the material. It makes more sense. These are the lobefin fish here, and the coelacanths, which were thought to be extinct for tens of millions of years, and they all of a sudden found them in very deep oceans. I think it was off the coast of Japan. The lungfish, the dipnoe, and then the Actinopterygii, these are the rayfin fish, and then the sharks, raisin skates. And then there's a little difference from one book to another on the name they give for the class for lampreys. And then here is the hackfish, the scavengers of the sea. And then here are some extinct groups here, the armor fish, the placodermi, and then the spiny fishes, the Canthodii and the Ostracoderms. Those are all, they're extinct here. These are the approximate numbers. You see the rayfin fish are the most abundant. Don't try to memorize numbers, but you should have some sense of understanding that the most diverse is the rayfin fish, the ones that we typically eat at the table. For the coelacanths, there's only 2, and they were recently discovered as still being alive. They'd only been seen in the fossil records. So looking at the first fishes, I got a picture of a lamprey. They are alive today. This is what a lamprey looks like. These are not necessarily teeth as we know them, but here's their mouth here. The gills are inside, and these little openings here where water goes in the mouth, then goes to the back of the pharynx, and then exits out after passing over the gills. Here's your tail fin in the back. No paired fins along the body there. And so we're going to call these Agnatha generally, the group. And that's because nathos means jaw. And when you put an A, that means without. So Agnathos, Agnatha, these are fish without jaws. These are the extant fish today. And they're separated into 2 classes, the Nixini, which are the hackfish. That's their class name. And then the class name for the lampreys is Cephalospidomorphi. But what I noticed in this book is they're going with a different name, and I don't know which one is the one that's most accepted. And so be aware of that even the notes still have the two different names. Now the one that your lab mate is going to go with, and the outline that I give you for the names to know, is cephalospidomorphy, but I include the one they have here. These are still alive today, and they share close ancestry with other jawless fish from those ancient oceans. The Ostracoderms found in the Ordovician and into the Devonian from 485 to 360 million years ago. And this group is now extinct, and I got some pictures of some of what they look like. The Ostracoderms means like bony skin type, but they don't have jaws. And excuse me, and they are in some cases scavengers and so on, and many not very big to begin with. But you can see this line of jawless fish. The current scheme seems to have split off before, and the common ancestry there before the other group of fish evolved from this common ancestor to both down here, right about there. And so this is what the former textbook had. They had cephalospidomorphy there instead of the Petiro. I haven't even tried to pronounce that name. And so we have to have jaws appear. So the early vertebrates didn't have jaws; that they were in the water like fish. And so, where did the early evolution of the jaw occur from? Well, we want to look here at, first of all, those early fish had these arch-like structures made of cartilage. And those gill arches help support the fleshy part of the body in the back of the pharynx to keep it from collapsing down on the gills. The gills are inside the body there. And so these gill arches are made of cartilage, and the gill arch starts to become modified. So these first gill arches start to become jaw-like structure, and then further modification to a jaw that is firmly connected to the rest of the head, and then to the shoulder, and we're allowed to open that mouth even wider, so you can take advantage of larger food items and become even more successful as a group. So another group that's on that table is the armored fishes called the placoderms. There's the ostracoderms, and then there's the placoderms, and then the spiny fishes was part of those. So the spiny fish belong to an extinct class called the Acanthodidae, and they were common during the Devonian. And so this is where I would go back to my timeline, and I'm starting to look, where's the Devonian? And then I start drawing in, where did the placoderms fit in? So that's why I had made an outline, because there's not one here pictured. And so this group replaced the ostracoderms to become more dominant. And during this time, the ostracoderms from the prior slide were still around. And then these were evolving, and then when they replace them is becoming more dominant in the oceans, more abundant, more diversity. The cartilage skeleton, but the skin had small plates of bone. And these guys became extinct at the close of the Permian. There was a massive extinction at the end of the Permian period. I forgot what the number was, like 95% of all species got wiped out on the planet during that mass extinction at the end of the Permian. It's called the Permian extinction. And so the fins were reinforced with spines, which gives them the name spiny fin. And then the placoderms, the class Placodermia is the name for this group. They became common in the mid to late Devonian. So we have fishes that are occurring together. And here the jaw was more improved than in the spiny fish. The upper jaw was going to be fused to the skull, and the skull hinge on the shoulder, and that again is going to allow for much wider gape for the mouth. And then these were extinct by the end of the Devonian. And here's some relative to the size of a human here. So this is a rather large fossil that was found and kind of outlined the way the silhouette, the way the fish might have looked. And then others quite small here, you can see relative to the size of a human hand. And so now we move on to sharks, which are known for having a skeleton made of mostly cartilage overall. So the sharks were around during the time of these other fishes. So we go back to that Devonian period, and you go back to that point, and A lot of those fish are now starting to be erased from life on this planet, and so many of them are disappearing. But these groups gave rise to other groups, right? And sharks had been around since way back. They've been around since the early Devonian, and so if we go back to some of these, they were around during the time of these. These guys were around in the Devonian as well. So we go back to 400 million years ago to see some of the first sharks, and the sharks had some major improvements on the jaw even further, so making them better able to outcompete some of those other predatory fish that were around at the time. And so again, we see the same drawings from earlier on the origin of the jaw itself, same concept. From those gill arches, we see them start to move and become parts of the jaw overall. So sharks, after they began to evolve, and here's where they are on, today we call them chondrichthyes. Chondros, the name is chondrichthyes. And ichthyos means fish in Greek, and chondros means cartilage. So literally translated, cartilaginous fish. So there's the name chondros, means cartilage. Ichthyos means fish. So the sharks were dominant sea predators by the time of the Carboniferous. So those other fish, this fish are now the kings in the ocean. And the chondrichthyes also include stingrays, rays, and skates, but they haven't evolved yet. They evolved later. So early on, it was just sharks, different groups of sharks. Again, their skeleton is cartilage mostly. There are some granules of calcium carbonate in there, but mostly cartilage, which is lighter than bone, yet pretty strong. As there's a picture of a shark right there. And so sharks have some teeth. We've come to know and understand. And so these sharks were among the first vertebrates to develop teeth, what we would call teeth. And they evolved from rough scales on the mouth and the skin. So not necessarily originating on the jawbone itself. So they were not set in the jaw. And for sharks, they easily lose their teeth, but they're constantly making more teeth. They don't necessarily have the same origin that our teeth would. So they have smaller teeth further back in their mouth, and as the older ones get lost, they are being replaced by the new teeth that are being produced. They do have a special sensory organ called a lateral line. Both the sharks and the bony fish we're going to see later, which are around, by the way, at the time and starting to evolve during the time the sharks are around. But this is a line along the side, the lateral. It's called the lateral line, and it has little pits in there, and within those pits are receptors that are sensitive to pressure changes. So basically, it's kind of like they're able to sense vibrations in the water when other fish are swimming around and so on. It's almost like hearing, but for underwater. And so they're able to detect pressure waves under there and respond to them. Sharks also have some very special electrical receptors in the skin up in the head region, as well as pretty interesting. Now for sharks, their reproduction. The sharks does differ from other fish in that the eggs are internally fertilized. In other fish, the female lays the eggs and the male comes and releases the sperm to fertilize the eggs. And that's not to say all other fish are not capable of internal fertilization. There's these little top minnows that are not sharks, rays, or skates. They're the ray fin fish that evolve, come to become more dominant a bit later. And there's others that have specialized fins that allow for internal fertilization. But for the most part, sharks, internal fertilization. Many pups are going to be born alive. There are a few species that do actually lay fertilized eggs, and the eggs are somewhat leathery, but for the most part, the embryos are retained inside the female, and then the young sharks called pups are born alive. Now, sharks do have a long gestation period. That's the period in which the embryonic development occurs before they're born, and that's a big problem. It's a problem if you have a long gestation period and you don't reproduce very many and very often. If your population declines, it's very difficult to recover for that. And a lot of sharks are threatened with becoming extinct, mostly due to human activity on the planet. And one of the things is capturing them as bycatch in these large commercial fishing boats, and they don't want them as part of the catch, so they end up, sharks need to swim to ventilate their gills, and so they have to keep moving to get water, so they may die just by being captured, even if they're in the water, before they get pulled out of the water. If not, they end up dying before they get kicked back into the water. And then this has been popular in, especially in parts of the world, in Asia and some of those cultures. It's the sort of status thing to have shark fin soup. Even though the shark fin doesn't taste like much, they must add spices or whatever, but eating it for some reason is some sort of status thing. And so there's a big financial incentive to go out there, catch lots and lots of sharks, cut off their fins, and don't even use the rest of the body. They just dump the dying shark into the water. They can't even swim, and they don't regrow their fins. And so a lot of the sharks are now threatened with extinction, and they are important. All ecosystems require these top-down control from predators. Otherwise, the ecosystem has lost an important component. So that's how shark fin soup is a problem, and it's related to the slow reproduction that those occur. Now, as far as shark evolution itself, just the sharks, there were a lot of different kinds of sharks in those oceans, and many of them died out. We mentioned the Permian extinction. Many died out at the end of the Permian, 248 million years ago, when there was that massive extinction. And it's associated with evidence that suggests there was massive volcanic activity on the planet, enough to cause climates to change and changes in the environments that a lot of these animals were living in. Now, the survivors, the sharks that did survive, they diversified in the Mesozoic era. The Mesozoic era is the age of the dinosaurs, and so this is when sharks are still out there in the ocean, and then your dinosaurs are up on land. And the Mesozoic, you might be familiar with the Triassic period. It's part of the Mesozoic. I know you heard of Jurassic because of the movies, and so these are there. And then the Cretaceous is next. And at the end of the Cretaceous, which is 65 million years ago, the dinosaurs go extinct. So the survivors diversified in this Mesozoic era, and the modern sharks that we see today appeared, and this is the time when skates and stingrays appeared. Here's a picture of a stingray there. So during the early shark evolution, there was no stingrays. They evolved during the Mesozoic era. And so now we move on to bony fish, and bony fish now dominate the water there. You can see I just had that little replacement there. The Pteromyzontida is one name for that class of lampreys, and then Cephalospidomorphi is the name that your lab manual has. So we were looking at the chondrichthyes earlier, and so now we're looking at bony fish. This one shouldn't even be highlighted. I'm not sure how I ended up with that one there. Oh, I know what I did. Anyway, now we're looking at these fish here, and all of these fish have bony skeleton. And then your amphibians are here. So we're not going to talk about amphibians yet. But these are our bony fish groups here. The ray fin fish, the fleshy fin fishes that are here. And I'm not sure whether they're using Actinista for the fleshy fin fish, and I'll mention that in a little bit. But anyway, these bony fish were around at the same time as the sharks 400 million years ago. They just took them a while to get going, I guess. So again, I go back to my timeline and I draw this in there to see the overlap so I can better understand what's going on. We have to keep going back in time. Now, the thing about the bony fish here is that they adopted a heavier skeleton. And sharks have a lighter skeleton, but sharks are still heavier than water. They're more dense, so they're going to sink. And they sink for two reasons. They'll go downward because they have an asymmetrical fin. The top part of their tail fin, their caudal fin they call it, is larger than the bottom one. So when they're moving that fin back and forth, it causes a downward thrust and causes them to move down. but their pectoral fins are like airplane wings, and then that helps keep them up in the water. So they have to keep swimming to stay up in the water and also to get water to go through their gills in their mouth. They can't actively pump the water the way bony fish can. And so how do the fish stay, these bony fish stay up in the water? Well, they have an extra adaptation that is internal in their body. Now, this is a very diverse group. Over 30,000 species have been identified. And the organ that helps keep them up in the water column instead of sinking is called a swim bladder. And then they also have these gill plates or gill covers. It's going to assist them to ventilate their gills so they don't have to keep swimming. And so taking a look at the swim bladder, if we go and do a dissection and go into the fish here, there's normally a gill plate covering or operculum covering those gills and the rest of the body there. But if we remove all of that, we look in there, you can see the backbone here, and right underneath there is that swim bladder. There's the other internal organs there, the intestines and the stomach and so on. Now, if we go and we look here at what the swim bladder looks like, there's a gas gland right there, and all the gas that fills that swim bladder comes from gases that are in the blood. Our blood is carrying oxygen, carrying carbon dioxide, carrying nitrogen gas in there. And so what happens here when the blood vessels go to that gas gland is able to extract gas from there and fill the bladder. And the more gas you put in there, the more buoyant you become. Buoyancy is the ability to float in a fluid. And they're able to move and change their levels based on that. And then they're able to remove it from the swim bladder slowly and get it out of there back into the blood. So that's their adaptation, and that they don't have to keep swimming. You can see I mentioned the sharks have an asymmetrical tail. These fish have a symmetrical tail, which allows for a great deal of agility in the water there. It allows for a lot more movement than what a shark is capable of doing. Another fine adaptation that gives them an advantage. And it creates just simply a forward thrust when they're swimming overall. And so this is interesting because this structure is starting to look something like what our lungs do. Our lungs fill with air. And so, in fact, there are some primitive fishes that they don't have a swim bladder, but they have an outpocketing. So I talked about the back of your mouth, your throat area is being called the pharynx. So food comes into your mouth, and in the back of your throat is the pharynx, and then there's the opening to a tube that leads to your stomach, and that tube is probably further back over here. The tube would be further back over here, and then the stomach out here. Those would be called the esophagus. And back here, where you normally would have your gills, this is the back of the throat here. This region back here is the pharynx. Okay. And so what happens here is that any food you take in goes through here, but if you start to get this outpocketing or pouch start to form or evolve here, and then the fish can now take in air and swallow it and fill that sac with air. So these air sacs, and now not only that, if you surround this with little tiny capillaries, little tiny blood vessels, those blood vessels can now pick up oxygen from that air you swallowed, and we have a rudimentary or very primitive lung-like structure. And for the bony fish, they're going to have these gill covers. These gill covers actually have a fancy name called operculum. And operculum is singular, so sort of a hard plate that covers there. And what we're doing is we're looking from above the fish here in this picture right here. And the opercular on either side, this is the rest of the body of the fish here. And so the fish now can, if we look inside the pharynx back here, the fish can take food and water into the oral cavity right up here and into the back of the pharynx here. And any food can be sent through the esophagus to the stomach here. Water can be taken in. So when the fish opens its mouth, it's going to close those plates on either side, those opercula. Those seal up, and then they can actually open them up, and there's a gap that forms there. But right now they're sealed. So when they open their mouth, that's going to lower the pressure in their mouth because the volume of their mouth is getting bigger. There's a pressure-volume relationship. that you might study in a chemistry class a long time ago. The water goes in because of the lower pressure. And then when they close their mouth, they have this little valve-like structure right here that doesn't allow water to go back the other way. And when they close their mouth, the pressure increases and the opercula open, and that causes the water to flow out the opercula, but not before passing over the gills. So they're able to actively ventilate their gills. So the next time you're at an aquarium, near an aquarium, look at those fish up close, and you're going to see they open their mouth and then they open their opercula. Open their mouth and open their opercula. Open their mouth, close, and then the opercula open. So they're constantly going through these cycles to help ventilate those lungs, and they can just be at rest in the water, just sitting there in the water column and get water to pass over their gills. Sharks cannot do that. Okay, so looking at the major groups of bony fish, we have the ray fin fish, and they really have these bony rays in their fins there, and they help provide support for those fins. These are the common fish we're used to. Most fish we're used to. They're in the class Actinopterygii, and you see that in that cladogram that we're showing the relationships with vertebrates. They don't have any muscles that actually control the movement of the fin itself. It's the base of the fin that's moved overall. And it's moved not because of any muscle within the fin itself. And then you have the lobe fin fish, and there's some difference in the cladogram we were looking at and the way the names are used. Sarcopterygii means, sarcos means flesh or meat. So sarco, and then the pterygii, the pteryg is a reference to the fin. So this is ray, actino means rays, and ray fin fish, and then this is fleshy fin, translation. Okay, so that's the class I'm more used to using. And then actinista is what we see in there being represented as a class if we go back to that cladogram that shows the phylogeny. So we're going to call it an SC for subclass. So actinista is a subclass for our coelacanths. And the lungfish is a subclass for the dipnoi, and these two would collectively be called the Sarcopterygii. So what they have represented on that cladogram are these subclades or groups that we have names dipnoi and actinista. And so we can see that right here. There's the, this is for the amphibia, but here's that cladogram here. Here's the dipnoi, which are the lungfish, and they have fleshy lobes for their lungs, and then actinista here, and together that group right there is the Sarcopterygii. And so they're considered the rank of subclass. So there's two coelacanths and 2 species and 6 species of lungfish overall, and so that's a distinction there. But they do have the opercula and the gill or the gill plates. Now, if we come in and we look at what's in the lobe, that fleshy lobe. So this is true of both the pectoral fins and the pelvic fins. You're going to see some bones inside the animal, which would later become the shoulder bones, and the shoulder girdle, like the collarbone that we have here. And if you look in that fleshy lobe there, there's both muscle and bones. And if you look at the bones, they have the same arrangement we do. We have one bone that's connected to the girdle bone, to the collarbone. We have one bone, that's our funny bone, our humerus, and then we have 2 bones in the forearm, the radius and the ulna. They have that same arrangement there.